Hello everyone, you are all welcome to this video on genomic imprinting. Genomic imprinting, this is a heritable disorder that is caused by the natural silencing of an allele by epigenetic factors. And the main epigenetic factor that is involved here is methylation. You remember when we did the video on epigenetic factors, we said methylation is the main epigenetic factor that is involved in silencing an allele or a gene. If you want to know more about these epigenetic factors or epigenetic traits, then you will look for that video on epigenetic factors. So we'll be answering the question, what is the difference between a recessive allele and an imprinted allele? Okay, let's consider small r to be a recessive allele and small t to be an imprinted allele. So if r is a recessive allele, it means that r cannot express itself in the presence of the dominant allele capital R. Okay, but R cannot express itself in the heterozygous form, but R can express itself in the homozygous form. Okay, but an imprinted allele small t will not express itself, be it in the heterozygous form or in the homozygous form, okay, because it is imprinted. So if it is in a double copy, it will not express itself, and even when it is in a single copy, it will not express itself. Okay, so to summarize, a recessive allele will express itself in the homozygous form, but an imprinted allele will not express itself, be it in the heterozygous or in the homozygous form. So we'll look at two examples of situations where we have genomic imprinting. The first one, we'll look at the Angelman syndrome. This is a disease that is generally characterized by sleep disturbance, frequent laughter or smiling, and usually a happy demeanor. Here, the paternal UBE3A allele, which is usually found on the longer arm of chromosome 15 between positions 11 to 13, is naturally imprinted, that is, it is naturally silenced, and the maternal allele is either mutated or deleted. Okay, so that is what leads to the Angelman syndrome. If we look at this, our diagram below here, in the Angelman syndrome, this allele, UBE3A allele, is naturally imprinted. Okay, the paternal UBE3A allele is naturally imprinted. Normally, in a normal individual, though at this level there will be no problem, because the maternal allele or the UBE3A allele that a normal individual inherits from the mother will produce the normal proteins. But in the Angelman syndrome, the paternal UBE3A allele is imprinted, and on the other hand, the maternal UBE3A allele is either mutated or deleted. In the case where it is the maternal allele is mutated, that means that an abnormal protein will be produced. And if an abnormal protein is produced, the normal functioning of that protein will not be available. Okay, so you produce an abnormal protein that will cause an abnormal situation in the system. But in the case where it is deleted, okay, no protein will even be produced at all. And in that case, there will be a deficiency in the normal protein and therefore an abnormal metabolic situation in the system. Okay, so that will lead to the Angelman syndrome. Now, the second case, we have the Prader-Willi syndrome. This is a total opposite to the Angelman syndrome. And here, this is a disease that is generally characterized by weak muscle tone, feeding difficulties, poor growth, delayed development, obesity, sleep abnormalities, and delayed or incomplete puberty. Here, the maternal Prader-Willi alleles also found on the longer arm of chromosome 15, still in the position between 11 to 13, are naturally silenced, and the paternal allele is either mutated or deleted. Okay, so it is a total opposite to the Angelman syndrome. In the Angelman syndrome, it is the paternal allele, the UBE3A allele, that is naturally imprinted. But in the Prader-Willi syndrome, it is the maternal Prader-Willi alleles that are naturally silenced or imprinted. So that is the effect that genomic imprinting can actually cause in the system. So thank you for following the video, and see you in the next video that will be uploaded.